Hi everyone, my name is Felipe, and today I'm going to present my research titled Molecular Dynamics and Machine Learning Review Variant Specific Residues of Spike TMP RSS2 Interaction in SARS-CoV-2. As we all know, SARS-CoV-2 had a global impact with over 700 million cases and 7 million deaths reported so far. This is a representation of the virus. The red structures you see here are the spike proteins which play a key role in the infection process by allowing the virus to enter human cells. This diagram shows how SARS-CoV-2 infects human cells. First, the virus binds to ACE2, then the spike protein is cleaved by two proteases, first bifurin and then TMP-RSS2. TMP-RSS2 is specifically important because its action helps the virus enter the human cell. That's why it's a key target for studying infection and possible treatments. Here are some of the SARS-CoV-2 variants. These variants appear due to mutations in the genome of the virus. Some variants like Delta and Omicron can affect how spike interacts with human proteins. So we want to understand these changes, specifically how the variants interact with TMP-RSS2. To study how spike interacts with receptors, we use molecular dynamic simulations. These simulations let us see how the different complexes behave over time and how it changes its conformation during interaction. But these simulations create a lot of data, which is hard to analyze manually. So we use machine learning to find patterns and differences between these variants. By combining both methods, we can better understand how each variant interacts with human receptors. To achieve this, we carefully model the spike protein from multiple variants, including the glycans present on both spike and ACE2. We then performed molecular docking to investigate interactions with host receptors, followed by molecular dynamic simulations to assess structural changes over time. Stability was evaluated through the RMSD over time, while hydrogen bonds and binding free energy were analyzed using the MMPBSA method. Finally, we applied a random forest model to identify the most relevant residue pairs involved in these interactions. The RMSD analysis of the spike TMP-RSS2 complexes revealed distinct behavior for the wild type Omicron B5 and B2 variants, possible due to differences in their interactions with TMP-RSS2. The remaining variants show greater overall stability throughout simulations. As global RMSD differences were subtle, we performed a more detailed analysis of specific interaction regions between proteins. In the complex with TMP-RSS2, we identified key spike residues, which are D796, E819, D867, and D868, which are interacting with important TMP-RSS2 residues such as E299, D345, and D465. A major hydrogen bond was observed between the residue K814 on spike and E299 on TMP-RSS2, indicating a potentially critical interaction for complex stability. However, this initial analysis did not allow us to clearly determine the impact of specific spike mutations on these interactions. For this reason, we proceed with a more detailed investigation using the machine learning. We applied random forest models to analyze 16,000 residue pairs between spike and TMP RSS2. Many of these pairs were conserved across variants, but most variants, except alpha, also present unique and important interactions. For instance, in delta, the pair 940 and 341 may be impacted by the nearby mutation D950N. In Omicron BA1, we identified the pair 952 and 336 located near the Q954H mutation. Additionally, the D796Y mutation may influence the pair 812 and 296. In BA2, the mutation D796Y appears again and may affect the pairs 796323, 793390, and 794446. In XBB.1.5 variant, the S939F mutation may contribute to interactions at 791, 347, and 792, 398. In BA286 variant, the S939F mutation directly forms the unique pair 939409. In conclusion, we identify key residues from both spike and TMP RSS2 involved in their interaction. Some of these residues are conserved, while others are variant specific, especially in Omicron. Our random forest analysis was essential to highlight the most important residue pairs. As a next step, we plan to study how genetic variations in human receptors or polymorphisms might affect the bind strength and stability of these complexes. This could help us better understand how mutations impact viral entry and may guide future therapeutic targets. Thank you, everyone.